Land plants possess a special set of cells that are perpetually alive, perpetually dividing. These are called meristems, and you can think of them like stem cells. They essentially live for the entire life of the plant. They make all of the other cells of the plant body. So every cell in a plant body eventually came from a plant meristem. And these meristems occur in very particular places. But what are meristems? What do they do? Here's a slide of the idea of a meristem. Plants grow from meristems. They are, as I say, the ultimate source of all plant parts of the mature sporophyte. They are undifferentiated. That is one of their major features. They are undifferentiated. They retain forever, in the sense of the duration of the plant's life, the ability to divide. And here I've drawn some cells. M means meristematic cell or meristem. And we start out with some meristematic cells, and then as we move along in time, in the middle, you can see that one of the meristematic cells has given rise to a daughter cell. And that daughter cell I've labeled D. And that daughter cell then can go on and differentiate and become something specialized. So on the far right, we have our meristematic cells, and then we have the daughter cell that's divided into further cells, and then will become some other mature cell type. The essential feature here is that the meristem remains. So when a meristematic cell divides, one daughter cell remains a meristematic cell, and the other daughter cell goes on and does something else. So we have a continual meristem, something that lasts the entire life of the plant. Where do these occur? Well, meristems come in a couple of types. There are apical meristems, and these occur in the roots and in the shoots. And you can see here in this diagram from your text that they are at the tips of shoots. So each shoot, whether it's the top of the plant or whether it's an axillary bud, has an apical meristem. And the roots, each growing tip of a root has an axillary meristem. This is primary growth. So the shoots and the roots grow by primary growth. That's the first stage of growth. Some plants become woody, and woody plants have another set of meristems, and those are called lateral meristems. We're going to discuss those in the next lecture. Today we're only going to talk about so-called primary meristems, the apical meristems. But the lateral meristems I'll mention now consist of a couple types, the vascular cambium and the cork cambium, and they cause what's called secondary growth, and that's associated with wood and with bark. But today we're talking about primary meristems. So here is a longitudinal section through a plant that you all know. This is Coleus, and Coleus is this plant from the mint family that has these variegated leaves and all sorts of neat colors. And if you take a shoot of a Coleus and make a longitudinal section, that's right down through from top to bottom, right through the apex of one of the shoots, you can look at this under a microscope with some stains and see the apical meristem. So here it is. You can see in the center at the top are a bunch of cells. This bunch of cells is the apical meristem, in this case the shoot. And all of the other cells came from that apical meristem. Try to wrap your head around this. Every cell that you're looking at came from the apical meristem that's sitting there in the center of this picture. This is just a walk through time. That's one really neat thing about plants, is you can look down through time in a plant and see the history of cell division. So you can see a young leaf on the right, it's labeled here, and that young leaf used to be cells from the apical meristem that have now differentiated and developed into leaf cells. And there are other cell types. There's a young developing set of vascular tissues in a strand. And then there are also axillary bud meristems. So if those axillary buds ever become shoots, they're in the axil of the leaf. If they ever become shoots, then that meristem is what's going to cause them to grow. All the cells in this picture came from the shoot apical meristem. So remember that there are three tissue systems in plants, the dermal, the vascular, and the ground. And these tissue systems arise from three new meristems that are made by the apical meristem. So in particular, the apical meristem makes the protoderm, so that's a meristem, and that protoderm makes the dermal tissue, that is the epidermis. A second type of meristem made by the apical meristem is the procambium. The procambium makes vascular tissue. The third type is called the ground meristem, and the ground meristem makes ground tissue. So let's draw a meristem. Let's draw a shoot apical meristem. Okay, so the shoot apical meristem is at the tip here. So that's the SAM. Shoot apical meristem. A group of cells. There will also be a shoot apical meristem right here at the axillary bud. If this ever grows, a shoot apical meristem will be the source of all its growth. As the shoot apical meristem produces cells, it makes these 3 extra meristems, these 3 new meristems. And the one that's on the outermost part of the plant is called the protoderm. So that would be here or here. So this could be called the protoderm. It's also producing cells that will become the vascular tissue. And those go like this down in the plant. They extend up into any leaves and up into any shoots. And that is called the procambium. All of the other material in the young plant, here, that surrounds the vascular tissue, or what will become the vascular tissue, is called the ground meristem. And this will produce the ground tissue. This is a leaf primordium. That simply means it's a small leaf that will develop into a larger leaf. So leaf primordium. And this is a slightly more mature leaf here. You could call it a primordium if you wanted to. And it will become a leaf later on. So there you have it. There is a shoot, a longitudinal section through a shoot showing the shoot apical meristem, the source of all the cells that you see here. Okay, this is what we just drew. So here you can see our shoot apical meristem, and there are other young tissues. All these young tissues came from the apical meristem, and then one of the 3 other types of meristems made, that is the protoderm, the procambium, or the ground. And how do you know what's what? Well, the youngest tissues will be protoderm if they're on the outside of the plant. That's that single cell layer on the outside of the plant. And then somewhere there is going to be vascular tissue, and that typically occurs in strands somewhere beneath the epidermis. And then surrounding the vascular tissue, beneath the epidermis, but surrounding the vascular tissue and making up the bulk of the young plant, is the ground tissue. So that's ground meristem that's making that. And the point at which the meristem actually becomes non-meristematic, becomes the actual tissue, say the ground tissue, is not easy to see here. Just realize that it's a ground meristem, a set of dividing tissues that makes the ground tissue. The procambium, dividing tissues that make the vascular tissue, the xylem and phloem, and the protoderm that makes the mature epidermal cells after dividing and dividing and dividing, and those cells maturing. Now, if you took your pet goldfish and you made a cut in its nose and then several cuts down through its body, you wouldn't really see a difference in age in those sections of your goldfish. However, if you took a young plant and made sections from the apical meristem down through the shoot, you would see a time series. You would go back in time. And that's what's shown here. You can see the youngest tissues at the shoot tip here. So look at the top. there's the apical meristem. And as you go back in time, as you go down through the plant, you can see the 3 meristems that were just recently made by this apical meristem. These are meristems, the protoderm, the ground meristem, and the procambium. They retain forever the ability to divide. They retain the ability to differentiate. And once they differentiate, they become these primary tissues, the epidermis, the vascular tissue, the ground tissue. And you can see here, I think this is a really nice diagram because you can see a longitudinal section and then the corresponding cross-section on the right. So once we've gone back in time and we've looked back in the chute, we've looked down away from the apical meristem a bit, and we've moved away from the protoderm and the procambium and the ground meristem, and we see the young tissues that have developed. What do they look like? Well, they look a little different in monocots from the way they look in eudicots. So let's draw a eudicot and a monocot stem. Let's do a cross-section. So let's draw a eudicot on the left. Here we are. So this is a eudicot. A eudicot young shoot has as its outermost layer, of course, the epidermis. And then it has a ring of vascular tissue. These groups of vascular tissue are called vascular bundles. Each one of these is a vascular bundle. And this vascular bundle contains within it both the xylem and the phloem. And in the middle here is procambium, which has given rise to these tissues. And typically, toward the outside is phloem. And toward the inside is xylem. This whole unit here that contains the xylem and the phloem is called a vascular bundle. So that's a eudicot. The material that surrounds the vascular tissue is called ground tissue. Okay, so let's draw a monocot. Monocots have a slightly different arrangement of the vascular tissue. So of course here we have the epidermis on the outside, and the vascular bundles of a monocot stem are scattered throughout the stem. So they don't form this nice ring like they do in the eudicots. So here's the vascular bundle. And within each vascular bundle, there is xylem and phloem once again tends to be toward the outside, and xylem tends to be toward the inside. And there can be a little bit of procambium in the middle that remains, and then usually just disappears with development. The ground tissue, of course, is all the material that's inside, that surrounds the vascular tissue, and each one of these little vascular bundles is shown over here. So that's it. Monocots have stems that have scattered vascular bundles, and eudicots have stems that have vascular bundles in a ring. Here's what we just drew. You can see that eudicots tend to have a ring of vascular tissue, whereas monocots tend to have scattered vascular tissue. Much of the stem is made up of ground tissue in both cases. In both cases, there is an epidermis on the outside, a single cell layer. And there is xylem and phloem in each vascular bundle. And one thing to notice here is that phloem occurs outside of xylem. This is a common feature in plants. It's almost universal, that in a vascular bundle or elsewhere in a plant, the phloem tends to occur more toward the outside than the xylem does. The xylem tends to occur more toward the inside. All right, so those are shoots. What about roots? What if we draw a root? What's going on with a root and its apical meristems? Okay, so let's draw a young growing root. We'll concentrate on the apical meristem and the tissues it makes. So here's the root. growing root. And at the tip of a root is a collection of dead cells called the root cap. There it is. Right behind the root cap are the cells that make up the meristem. So this is the root apical meristem. There it is. This root apical meristem is producing the three meristems that we've talked about. So it's producing the procambium. And in the case of roots, this procambium extends down the center of the root. There it is. That is procambium. That procambium will become the xylem and phloem as it matures. Outside of the procambium is the material that will become the ground tissue. So this is called, at a very young stage, the ground meristem. And of course, the outermost part of the young root is the protoderm. Right there. And that's it. That is a young root showing the apical meristem and the three meristems it produces. The central part is where the vascular tissues are, and that region is called the stele. This is what we just drew. You can see a root cap. This root cap protects the root as it moves through the soil. And behind the root cap is the root apical meristem. And then all of those primary meristems, those three primary meristems, that will go on to make the mature plant body. So that's the procambium, the protoderm, and the ground meristem. Now roots move through soil. Roots have to somehow push through little crevices in rocks. They have to push by sand grains, through hard bits of rocky material, sharp materials. They're always pushing, and that's what the root cap's function is. It protects the nice soft plant body from all of these hard materials. And roots also have root hairs. These root hairs greatly expand the water absorption capabilities of these roots. And so roots have a very special way of growing. They have what are called three different zones. So if you look here at this plant, at the bottom is the root cap and then the apical meristem, the root apical meristem. And that's dividing. It's actively dividing, the apical meristem. And right behind it, the cells of the three meristems are actively dividing. Okay, so that's a lot of cell division. And after those cells have divided, then they elongate. And that's called the zone of elongation. And a little bit later, they make root hairs. And you can see that's called the zone of maturation. It's in this zone that root hairs are made. So in this zone, the root is no longer pushing through the soil. And this is really cool. This means that the plant has figured out a way to make its root hairs only after it stopped pushing through the soil. So the root hairs don't get sloughed off. They don't get shaved off. They stay there. And each root hair is a single epidermal cell, a single highly specialized epidermal cell that greatly expands the water absorbing capabilities of the root. So here's another view. This shows the zone of maturation or differentiation, same thing. Zone of elongation just ahead of it, and even younger still is the zone of cell division. So once again, just like in a shoot, you can take sections through a root and go back in time, from youngest to slightly older to slightly older. Here's a movie. This is a root growing through some agar, and you can see here it's pushing through, so there's cell division on the left, and then you can see that there are cells in the center in the zone of elongation that are elongating. And now look at the back. Look at the root hairs emerging. Those root hairs are not moving along. That's really cool. Those root hairs are not being subjected to the roughness in the soil. They're not being shaved off. They are emerging in a part of the plant that stopped moving. Here's another movie. Here we go. This is a root growing along. And now you can see the root hairs The root hairs are not moving down. They just emerge and they are nice and protected. There it is again. Here's yet another movie. These red dots are nuclei in the cells. And once again, you can see these are moving around. And this movie shows a lateral root emerging. And so in just a moment here, you're going to see how a lateral root comes off of a root. And there it is. And that lateral root, of course, itself has to have apical meristem cells in it. And it has it at just near the tip, right behind the root cap. And there it is. Off it goes. And that's a root branching. And you can see in the center there is material that you'll soon find out is vascular tissue. There it is. And the root hairs emerge, and they do not move after they've emerged. So the root hairs greatly increase the surface area of plants. And you can see here a 4-month-old grass plant. This is a rye plant. It has 14 billion root hairs. They provide a surface area of 400 square meters. If we were in the great big lecture hall in the Ondaatje Auditorium today, it would be about the size of that floor. 400 square meters is 20 meters by 20 meters. Place end to end, they would extend for 10,000 kilometers. So root hairs are really, really, really expanding the absorptive capabilities of the plant. Here you can see root hairs extending, and as I say, these are single cells, highly modified, highly specialized epidermal cells. There they are. So let's draw a young root just after the apical meristems have made their tissues, and the young tissues are dividing into the various types, the ground tissues and the vascular tissues and the epidermis. Let's make a drawing of that. So what do roots look like just after the apical meristem has produced the 3 other meristems, and they're very young? Let's go back a little bit beyond the apical meristem and see what young roots look like. So here we are. These are roots. And we will draw 2 cross sections, just like we did for stems. So over here, let's draw a eudicot. And in a eudicot, the outermost layer is, of course, the epidermis. Some of these cells will be root hairs. Inside is a very special layer called the endodermis. And this is 1 cell layer thick. I'm exaggerating its width here just so we can see it. So there it is. This is the endodermis. And the endodermis serves to keep material outside of the vascular tissues. And we'll talk about that later when we talk about vascular tissues and how water and minerals move up plants. Inside are the vascular tissues. So in the case of eudicots, we have xylem that occurs in sort of an X-shaped structure, X-shaped arrangement. So that's the xylem. And just outside the xylem is the material that is the phloem, that is to say the sieve tube elements. So that's the phloem. And just inside the endodermis is the pericycle. And those cells are cells that will give rise to any lateral roots that are going to emerge and come out sideways in the plane of this diagram. This material is called cortex, and it's ground tissue. Okay, so that is a eudicot root. What about a monocot root? Let's draw a monocot root. So here we are. Of course, the epidermis is the outermost part. There is, of course, an endodermis. Once again, 1 cell layer thick that I am exaggerating the width of here. There it is, the endodermis. The xylem and the phloem tend to occur in scattered vascular bundles. So each vascular bundle is relatively small, just scattered about. And the xylem tends to occur toward the inside, and the phloem tends to occur toward the outside in each vascular bundle. Here, the cortex is this material outside, and typically in monocots, you call the material inside the pith. That's ground tissue. Both cortex and pith are ground tissue. Now, you can see that there's an endodermis in the roots, and inside the endodermis is the vascular tissue. So the vascular tissue is protected from the rest of the plant. So here we have a root hair that might be on the outside of a plant. So here, for example, outside of a root is a root hair, and any material that comes into the plant through the root hair would have to travel across the cortex to get to the endodermis. And the endodermis then can decide whether it's going to let material in or not. And we're going to discuss that in a different lecture later on. So the stele is everything inside the endodermis. The stele arises from procambium. Okay, so here's what we just drew. You can see here the eudicot and the monocot organization of roots. Of course, on the outside is the epidermis. Inside that is a region called the cortex. And inside that is a very special ring of tissue called the endodermis. The endodermis is the innermost layer of the cortex. And then inside that is the stele. The stele is all the vascular tissue in the root and in the stem, and it arises from the procambium. There's xylem, there's phloem, and that occurs inside the stele. That xylem and phloem is inside the stele, which is surrounded by the endodermis. Notice that in a eudicot, the xylem makes an X-shaped, and that's really convenient because xylem starts with X. So think of a eudicot as having an X-shaped xylem in its center. If you see a picture, a cross-section, where you have a circle representing the endodermis and then an X, that is almost certainly a eudicot root. And then the phloem is just outside that xylem material in the eudicot root. In the monocot, the xylem and the phloem occur as separate little bundles, separate little regions, scattered throughout the center of the monocot root. So here's a close-up of xylem and phloem that shows the sort of X shape that I just talked about. And you can see the endodermis. And there's also a very special region inside the roots of these plants called the pericycle. And the pericycle is inside the endodermis, and its role is to provide the source material for a lateral root. So if a lateral root is going to emerge from this root, it's going to grow out in the plane of this slide, grow out radially, and it's going to start from the pericycle, these special cells that can still divide. So here's just another way to look at it, a eudicot and a monocot, and you can see the characteristic X shape of the xylem in the eudicot and the scattered xylem and phloem in the monocot. Here's how a lateral root emerges. It emerges from the pericycle that is just inside the endodermis. So here's a summary of what we've talked about in root growth of non-woody plants. So primary growth, that is. So there's a root cap. There are root hairs that are epidermal cells. The endodermis surrounds the vascular cylinder. It's the innermost layer of the cortex. It is outside the vascular cylinder. The vascular cylinder is sometimes called the stele, S-T-E-L-E. You'll see that word. It's in the center of the root. It contains the xylem and the phloem, that is the vascular tissues, and it also contains some ground tissue. Finally, there's this special set of cells called the pericycle, which lies underneath the endodermis and outside the vascular tissue, and it is the origin of lateral roots. Next time, we're going to talk about how plants become woody. Woody plants can get much taller than non-woody plants. They're stiffer, they're stronger, they're the source of wood, and they're the plants that make true bark. See you then.